cell phone, the current one version, and then next year there's another version of the cell phone, and then it does a little bit more. Well, that's the way some of the instrumentation is in the lab too. It keeps evolving and changing. We have a particular version of an electronic cell counter. It's made by a company called Coulter, so it's a black box. So what does it have in there? It has a door on the side here, and you put your sample in a plastic cuvette. So it's a little plastic vial, and you squirt some fluid into your plastic vial. We have a pump delivery system, big container over here. So you're going to squirt some of this fluid in here. This measuring thing here is set. This is already set to 9.8 mils. And you're going to take some of this cell suspension and you're going to add it to this vial. And you're going to actually add exactly 0.2 mils. So the total is going to be 10 mils. Total volume in your vial that you're going to be putting into the electronic counter. So there's a dilution factor here. How much is the dilution? If you put 0.2 mils into 9.8 mils for a total of 10 mils. Boy, it is Friday morning. Some people are still digesting their breakfast. Other people haven't gotten conscious yet for some other reason. Okay, so there's a 50-fold dilution, right? So do you need to multiply by 50? Well, no, because this is an electronic box, right? This is the Coulter counter. So it's got a little computer inside of it. So we programmed in that there's a dilution of 50. So the number it's going to give you a cell per mil number. It can never give you more than that because it doesn't know how many mils you have, and it could vary. In each experiment, you start with a plate of cells, and you might trypsinize with 2 mils instead. It doesn't know that unless you tell it, right? So, it's going to give you cells per mil, and so then you need to still calculate cells. So let's say you get a number off this machine, and it is that. So you have 51,003 cells per mil. Well, that's comparable to what you had up here. Did I do that right? No. 510,000. Okay, so you get this number off the Coulter counter. Well, that means that these numbers are pretty comparable. Does everybody follow that? So if I asked you, in the quiz about cell counting, you'll be able to tell me about the hemocytometer and why you can count the cells. You can count the cells because that slide created a specific volume, which was 0.1 cubic centimeter. So, 1 times 10 to the minus 1. And then the dimensions of each square, so you're able to calculate how many cells are in that field of 0.1 cubic centimeter. Okay, and then for the electronic cell counter, the number that it gives you is the number. We're all done, right? Almost we're all done. You still need to multiply times the number of mils, right? So that's the magic number. So if I ask you that on a question, how many total cells were in the sample? And the electronic counter says 510,003. So how many cells are there total? Well, you still need to multiply by 3 to get that magic number. So this Coulter counter is kind of new to us. I mean, we brought it out. It's been sitting underneath the bench for a while. But we recently learned how to keep it up and running a little bit better by rinsing it with a detergent solution when it's not in use and that kind of thing. So we're going to try to keep that going from now on. So that's kind of one of our objectives. We like it because it's actually, this number is a bit better than what you can get with the hemocytometer. we trust it more, and it's going to be more reproducible. So if you want quality numbers for your growth curves, I think this is going to be the winner in the long term. In the short term, there might be a few moments where we struggle with it, but I think it's going to work out for us. Okay, so now we don't have that much more time, but I did want to talk about the lab itself. You know, besides counting cells, and you know, what are these cells that we work with in our lab? What is our lab all about? Brandy, what is our lab?
Well, we work with thermal fibroblasts and we work with thermal fibroblasts.
Okay, what are thermal fibroblasts good at doing?
Reproducing skin cells and forming a skin layer of the epidermis and
Okay, well, the epidermis is a different cell type. And that's actually the thing that I wanted to talk about. So we tend to work on skin cells, number one, in our lab, because I have an academic background in the dermatology setting. And I like that as a model system. I think it's a great model system. You know, biologically, you know, how does that relate to the overall organism? You know, skin, you know, why am I in a lab where they work with skin cells? I mean, there's other interesting things there. Well, I mean, number one, the skin is an interesting organ. It's the thing that's most likely to get damaged in your life. You know, you'll get a couple scrapes and scuffs and, you know, perhaps some burns and maybe a couple of incisional wounds, you know. And then, you know, the quality of your skin kind of affects the quality of your life a bit and that kind of thing. So skin itself is important. It's a beauty item as well, so there's a cosmetic aspect to that. You know, but the structure of the skin, I like to generalize a lot. The structure of the skin is pretty universal, and that is, there's a layer, there's the epidermis and the dermis. So those are the terms that are used specifically for skin. But the epidermis is an epithelium. So this is a more general term. And the dermis is a mesenchymal layer, or a stromal layer. Sometimes pronounced mesenchymal. You go to these conferences and half the people are from different countries, and so, you know, some of them are saying mesenchymal, some of them are saying mesenchymal. One of the things about biology in general, which I always tell people, is there's a lot of redundancy. I think I might have even said that in the rotation. You know, you're going to be learning these terms, and there's a lot of overlap between the terms. Sometimes they're complete synonyms, but they were, you know, described by different labs studying different aspects of something. So one lab calls it one thing and another something else, and it turns out that they're total synonyms. They're talking about the same thing. And other times it's a subset. So the epidermis is a very specific type of epithelium. It's the skin epithelium. But there's epithelia elsewhere. Where else in your body do you think there's an epithelium? An example. Have you ever heard of an epithelia somewhere else in your body?
your lungs.
In your lungs. Yeah, there's a lung epithelium. It's good. So the lungs are on the inside of your body. So the idea of the epithelia is it's a layer of cells facing the outside. So why is the inside of your lung the outside? Yeah, I mean, it's exposed to the outside, right? You're taking in your deep breath, so it's the outside air, and sure, it has to go down, you know, down your nostrils or whatever, but it's going to go into the lung. And the cells that are facing that air as it comes in, they're actually facing the outside world. There's this air rushing in from the outside. So that needs to be an epithelia. There's going to be gas exchange occurring across an epithelia. And the thing that epithelia is always paired with is mesenchymal cells. So, you know, this is a general theme in biology that you've got two layers. Why is that? Why don't we just have one layer? Probably goes back to evolution, right? The idea that there was originally, a colony of cells, and there was a, one cell layer thick, and they joined together, and they formed some sort of wall or whatever. But that can invaginate into, something that looks like that. You've seen this sort of discussion, right, in your high school bio books and stuff. Simple life forms becoming more complicated, and one simple body plan is to, just fold the thing in, and once you fold it in, then there's the cells on the outside, and they're facing something a little bit different than the cells that are kind of nestled inside. So over time, they diverge a bit in their function. And so then you've got these two different layers, two different cell layers. So you've got the epithelia on the outside, and you have a mesenchymal layer on the inside. The classic embryological thing, though, is actually 3 layers, right? There's the like an epithelial layer, and a mesoderm, and an endoderm, right? So there's 4 different layers. So anyway, but this multiple layer thing, where you've got one cell type forming a tissue, actually, right? That's what a tissue is. It's one cell type that forms a structure, and then it's next to another tissue, and that tissue is the mesenchymal layer. Sometimes they say stromal, just to be biologists and confuse everybody. Those are really synonyms, actually. So I think the interesting thing about our lab is we focus on both epithelia and mesenchymal layers, and so some of the lessons we learn there about how those tissues interact. It's true for the lung. It's true for your kidneys. It's true for the organ systems through your bodies. It's true even for blood vessels. There are cells that are going to face the outside world and those that are facing the inside. On a blood vessel, which cell is facing the inside, by the way? Here's a picture of a blood vessel. It's a tube. So the cells on the inside facing where the blood is flowing. There's blood here. And then there's cells that make the outside of the tube. So which cells are the epithelia? The ones that are facing the blood, and the ones that are facing the You think that's the epithelia? Does everybody agree with that? And then the cells on the inside of the tube would be the zinc. Is everybody happy? You are? Well, the thing is about the cells that are on the outside, even though it's the outside, they're in your body, right? They're protected by your body. There's no blood vessel sort of hanging out in the outside world. It's not exposed to the air, right? It's nestled inside of your body. So it's actually the blood. In blood, they talk about blood having a salt concentration similar to the ocean. So it's kind of this fluid. These primitive organisms were used to seeing fluid, and there was this fluid with a bunch of salts in it and stuff. And then as these organisms became bigger, they needed to have fluid also kind of flowing around. And so the fluid was transported in these vessels. And so this fluid is actually the outside world. It's the outside seawater equivalent, further enhanced, certainly. But it's that idea. So it's actually the other way around. So I would say that the epithelia is on the inside. So they're called endothelial cells. Inside of the blood vessel. Anyway, I don't need to go into huge detail on that. Nonetheless, to give you some perspective, how long have people been thinking like this? Here's a book. I found this, the library was throwing it away. Boy, it smells too. It's from 19... Actually, it's not that bad. 1968. That's newer than I thought. I was going to say 1948. then here's the author. Johannes Hofstadter. So, you know, this book was just about epithelia mesenchyme. I mean, they looked in the microscope, they took pictures, they tried to document what's going on, all sorts of pictures like that. You've seen that kind of picture in a book. You know, they tried to understand the way the body plan of the works for all sorts of different organisms, and they took pictures of their observations and tried to understand what's going on. You know, you guys are so lucky because we're so advanced compared to this. I mean, these guys could only observe what was going on. Now we've got these very specific tools. So we have advanced microscopes, and we can, you know, take digital images. That's pretty cool. But that's kind of an enhancement of just a picture, right? I mean, the pictures are better. You know, there's another picture. I mean, now they're in color. You know, resolution is better, and you can see them on your iPhone and all that kind of thing. But that's still a picture. You know, what we can do beyond that is we can use antibody reagents to pick up certain proteins that we know that are in that section of tissue somehow, and so we can track where those things are. But there's all sorts of other tools. There's molecular tools today. So you can look at the proteins that are in there. You can look at the RNA expression levels that are reflecting which genes are being turned on and off inside the cell. So the tools that we have now are much better. And we're going to introduce you to some of those tools during this semester. So that's one way to look at it. And you're going to be following in this strong tradition here of epithelial mesenchymal interactions, and realize that it relates to the skin, but it relates to all sorts of internal organs. So it's a very fundamental biological concept. Any questions? You guys ready to do something in the lab? Okay, so today we're gonna do the initial thing for you, just because it doesn't make sense to have a lot of trypsinized plates, because we want to provide everybody with the same cell count, basically. So Than is going to do a digestion of some fibroblasts, and then you're going to be trying to count those in hemocytometer and also in the culture counter. And I think she's going to help you with the with the culture counter, and I'm going to help you guys with the hemocytometer. So we'll be shuffling groups around like that. Now, at the same time, we also like the idea of showing you the fancy imaging. So you have some experience with that, having rotated through the lab. But I don't think we did a movie. We got a movie together? You guys rotated? Okay, so we've got a camera which can take, what is it called, interval pictures, you know, which is really time-lapse. So it's going to take a series of still pictures, but it's going to take like almost 1,000 of them, like 900 and something, whatever, pictures. And so if we take that stack of pictures and we feed it to a program like QuickTime, it will compile the pictures and flash through them all in, 30 seconds. And so it will look like things are moving, and that's the way a movie's made anyway. It's a series of still pictures, right? And they keep changing a little bit, and so you see the motion of something. So, so we can make a movie. Typically, human cells, they move pretty slowly a lot of the time. So something like time-lapse photography could be really useful. Nonetheless, I mean, in this class time, we don't really have enough time to look at some of those interesting things. But we thought we would trypsinize the cells. And so when you're trypsinizing the cells, they're as flat as a monolayer. And as they're trypsinized, they're going to start to come off. And you're going to see that process of how they start to peel off the plate. And we're actually going to compare two different cell types. What two cell types are we going to compare? What do you think? What? Close. These guys. We're gonna look at epithelial cells and mesenchymal cells. And the epithelial cell is called a keratinocyte. So the cell type is actually, that's a bracket. So it's a keratinocyte. The word cyte is typical for cell, right? And keratin, well, skin cells are making keratin. And you think, well, when have I heard of keratin before? Doesn't a hair have keratin? Yeah, And the hair is a skin appendage. So, I mean, if you learn about skin, you're also learning about hair. You're learning about sweat glands, you're learning about melanocytes. There's a bunch of things. It's actually a pretty complicated organ. There's a bunch of contributors to that. Anyway, so you have the keratinocytes, and the mesenchymal are fibroblasts. So, you're here.
Back, yeah.
No. No, I don't want you to come back. Okay, we have this special treat today. This is Chris Endicott. You've seen him around? He's our head lab manager guy, and he's here to tell us about lab safety. And I invited him in. I think this is a real important aspect of the lab. So, you know, you could potentially get hurt in the lab. There's a few different scenarios, and you know, we want you to know how to handle that if something were to come up, and it's for your own protection. So, with that, Chris, you want to tell them?
Raise your hand if you've heard this talk in the last week.
Oh,
There we
everybody's
go.
heard it.
All right.
Oh,
So
no.
I'm going
So
to
you
do
don't
a
need
short,
to-
short version.
Okay.
So, and you're all just starting research methodology, right? This is 2990.
No, this is
This is
499.
490. So you've already gone through this a little.
You guys referred this for what? For chemistry or for? Yeah.
Yeah. Okay. Okay. So
What's
like
different
I said,
about
we
bio
don't
lab?
really need to go through this too much. That
Okay.
well, not so much. What's going to be changed is what the specific hazards related to this research in particular. And so you're going to be doing a lot of these labs in your research lab, right? 223. Okay. So there's going to be some specific things related to instrumentation and waste disposal and those kinds of things that Dr. Hoffler will instruct you as you get to those pieces. But you basically all know about the no food drinks and the not running around and the taking care of equipment and all that and where you're supposed to go in case there's a fire. So we don't need to do any of that stuff. We recommend safety.
Is that true? Is everyone's
because-
clear on that? You sign off on that? That you know what to do? Okay.
Yeah, no, I know you guys have heard it a bunch of times. Just to emphasize that when something goes wrong in lab, it goes wrong really fast. Okay, and that's why you need to definitely be prepared ahead of time. You know, that's why you should be already wearing your lab coat or already wearing your goggles if you think something's going to be dangerous. You know, don't reach into the sharp pins, things like that. You know, I dropped it. You need to be a little more careful that way. And you're going to be dealing with some other things now that are not typical lab situations. So you do need to worry about your own health, probably. If you cut yourself with something that's contaminated or whatnot, that's probably more your concerns at this point. If you a minor cut with something that you think is biologically dangerous at this point, you should rinse it with soap and water right away. And if it's particularly dangerous, you should definitely go get help. Like, don't wait that long. You know, get a Band-Aid on it and get out. Someone let us know. You also might be expected to come in at off hours and random times to, you know, passenger cells or split cells or do those kinds of things too. You should have somebody know where you are, you know, in case you don't come back at a certain time, you know, someone can actually check on you, work repairs and labs, that kind of stuff. This class is more of an independent thing. You know, this is not a regular class, right? You're going to need to be a little independent. You're going to be responsible. You're going to stay on top of what you need to do. But it is better not to be alone in labs. It's just super chill.
Okay.
The materials, you guys aren't really that particularly dangerous, you know.
Right.
I know. In one thing in your lab, it's a little bit congested, it's a little bit crowded. So try not to get yourself blocked into a bad place where you can't get out. Like here, you know, there's exit areas all over the place, but you know, keep that in mind.
Yeah, that's a
Make
good
sure
point.
there's a way
In the
that you
lab
know where
room,
you
there's,
are.
in every room, there's supposed to be 2 exits.
Yeah.
And so in our lab, even though it's a small lab, there's the outside door, but there's also a door that empties into the next door lab.
And it's there for fire. That's the only reason.
Yeah, so you're allowed to walk through that door. I mean, if you had to quickly evacuate our lab room, you could walk through that door to the next door lab, and then you can exit through that exit door. So there are 2 exits.
I'm just trying to think of anything related to your lab in particular that might be a problem. I don't think you guys don't do particularly dangerous chemical stuff. You don't have a lot of biologically active stuff. You're not doing doing pressurized stuff. So
Yeah, I think that's one of the reasons why doctors prefer going into dermatology, why there's so many dermatologists. It's a little less, dangerous maybe, but nonetheless, I mean, they are human cells, right? So this is probably the biggest danger, is that human cells could have a human virus in particular. And there's nothing I could really do about that. All human cells have viruses, and every human sample should be treated as contaminated. So if you're dealing with a piece of skin, you should treat it as an infectious agent. Now, there's possibility for infection everywhere, including the cafeteria and the handrails and the doorknobs and all sorts of things, right? But I mean, you have to be especially careful when you're dealing with cells, because if there were like a blood-related virus like hepatitis or something, I mean, we do everything we can to avoid that, but in theory, it's always possible that something will have some sort of virus in it.
So using gloves,
Yeah,
lots
so-
of times in labs, that kind of thing. And do you know how to remove gloves without contaminating yourself? Have you ever seen that one? So you have your gloves on, right? You take your glove hand and you peel it back a little bit so you can pull it out. Before you take your glove off, you slip your other fingers underneath. You use that to pull them off, so you're never actually touching the outside of the glove with your hands. Quite demonstrating. Okay, but yeah, I mean, things like that, you should just sort of be very conscientious about contamination and your exposure in this kind of class. And right next door to you, they're doing malaria research too, so they're already very aware of this kind of stuff. And, that is the live pathogen that they work with, but you have to be actually severely cut or having infected injected. So, there really are some interesting but dangerous things that we do here. So, this is sort of like the next step up for you. This is not just a regular sort of hand-fed class prepared lab, which is follow these instructions. This is really up to you to determine what needs to be done, what is interesting to look at, where you're going with this, and how best to achieve it in a proper way. You have to really worry about how you're sampling things, how your data collection, are you recording all your information properly. These are some of the more concerns you're going to be worrying about.
What's the policy on lab coats? So you mentioned lab coats.
Yeah, most people, there are some classes where they're required, and so you probably already have them, right? If you have them, you might as well bring them into this class too. This is a good idea where you just get in that habit. Those type of things.
Yeah, we'll sometimes decontaminate our cultures, and we use a bleach solution. So whenever there's bleach around, there's a possibility of getting a few drops on your clothes. And I've got several pairs of dark-colored slacks that have little spots on them. So and it's just from that getting through the lab coat or not wearing the lab coat when I should have it on. So, I mean, if you value your clothing, that would be something that it's a good idea to bring your lab coat and wear it. The
Everybody
department
knows.
likes that, actually. They like to see the students in the lab coats. If you want to, one of the things that we were saying last night, Natasha Khan gave a presentation. She did a very nice job. We were talking about like you package what your presentation is. I mean, each student, creates a sort of an image of yourself. We get to understand that you're a conscientious student, that that you're well prepared, that you study hard, and all these things. And what lab coat is a cheap thing that you can do that gives you a more professional image. So I mean, look at it like that. I mean, I'm not going to tell you absolutely can't work in my lab without a lab coat on, because to be honest, there's always a lot of people that are not in lab coats. We've got a couple of them in the lab now. Don't leave with any of our lab coats, but you're always welcome to grab one of our lab coats if it's sitting there and you feel like you need a lab coat. But you should also, I think, considering the fact that it's generally useful in labs, it's probably a worthwhile thing to have. Just get one.
And I know you guys have heard the whole thing about sandals, right? I see people in here that have already forgotten said thing, because you're now in the classrooms, and something on the last slide. So, you know, these are the kind of things. You got good habits, right? You got to get in the mode of doing this. This class here may be one of the most important classes you'll take here in actually. The undergraduate research is a huge benefit to students. You might not realize yet until you start talking to other students later on, you know, other colleagues who may not have had this as an undergraduate. It makes a huge difference on your career and what you can do. So, you know, treat it seriously, treat it professionally, and you can actually get surprisingly a lot out of this. I did a similar thing when I was an undergraduate. I was in the laser lab at that point, and it got me very interested in spectroscopy, and that sort of determined the rest of my career. So, it's a very good opportunity here to take advantage of it, but it is tailor-made, so for specific projects. And you need to learn that quickly and you know, be on top of it, start treating it like a job almost, or whatever it is. And identify things ahead of time. You know, learn what the chemicals are you're using ahead of time. These type of processes that deal with robots.
Great. Really, we're out of running on Friday, but okay, so we're just about to go into the lab, but so we talked about what we're going to do. We're going to count cells. It's kind of a tool that we need for what we're going to be doing in subsequent weeks. And there's two different methods you're going to learn. And you need to be able to tell me what the total number of cells is, not just the cells per mil. And then the other thing was we're going to make a movie. And the movie is of both keratinocyte culture and a fibroblast culture. And we're going to add trypsin EDTA to them, and the cells are going to start to peel off the plate. And you've seen that before, right? But what's the purpose of this lab? Remember I said that on quizzes, I'd love to ask, well, what was the reason we did that? We did something, we saw something, conclude something, so, you know, what's the lesson that you learned from that? And we're comparing these two different cell types. So the key is going to be there's something about the comparison between what you see in an epithelial cell when it's trypsinized versus what you see in a mesenchymal cell, these fibroblasts, what happens when they trypsinize. So I don't want to tell you what that is, because I want you to observe it. But when you observe those two movies, see the differences, the way the cells kind of look as they come off. You know, are they connected or not connected? And, you know, how do they look as they trypsinize? So that's going to be a key concept here. Okay, so I think we're ready. Let's go to the other one.